So I'm going to talk a bit about this kind of new analysis, the Amelogenin analysis of dental samples. And so let's go into it. These results that I'm going to talk today are part of this project that I'm developing now is in the last year. So we are trying to test if we can distinguish biological sex by looking into this protein that is in the enamel of the teeth, because there are many questions about the sex of the prey and the issue of selection of prey, as we've seen in this today in some talks, right? We are very interested in all this processing of hunting strategies, human behavior, if we can track more in detail what they were hunting and in which season, and if they were hunting preferentially males or females, because it has to be also with this protein acquisition, energy, and all that different questions, right? And besides that, in my project, I'm also combining it with lipid analysis from the sediments where these remains are, so we can put these remains in their paleoenvironmental context, right? We can look at the different vegetation types, and also through stable isotopes, we can check about temperature and humidity. So the project is quite big. It started only being from the Eastern Iberia, but now there's many people that wanted to collaborate with their samples. So now I have 140 samples from 6 different archaeological sites. And of course, we are also making a reference collection of modern European ungulates and carnivores that we can compare these proteins to the archaeological ones. And today I'm going to show only the results, or preliminary results, I should say, from the salt. This is a Middle Paleolithic site, and the remains are in the layer that are dated to 52,000 BP. So a bit of pain, but bear with me. So most of you might have heard or know about the SUMS analysis. So this is the protein analysis that we do for identifying different species when we have very minimal bone size remains, or when the DNA is not well preserved. That happens a lot in Pleistocene periods. So while checking at different collagen peptides, we can check the differences in the sequences and their mass. And if you see here in the screen, these little letters here are the different amino acids. And when they change, we can detect different species because it varies with the species, families, and species. And the Amelogenin works in a similar way. We cannot differentiate between species of taxa, but we can differentiate when these amino acids and the peptides changes, that usually happens here in this middle part of the gene. We can differentiate between male and female, right? Because the dental enamel produces this protein that is called amelogenin, and this amelogenin has X and Y chromosomes in it. So if you don't have DNA, but we have the proteins, we can still know more about this individual. This technique was developed in forensic sciences some years ago, especially for identifying human remains. In archaeology, has been used for the past 5 years to human remains, but now we are starting also to apply it to animals. So it's been very funny to now. Every time that I look to an animal, I just look at their teeth. And I just want to sample the enamel to have more referential material. So what we usually do is very straightforward. We have the protein when we got it from the sample in this way. So we needed to deconvolute that, open it, because what we need is the amino acid chain. So we need to use some enzymes to, like the trypsin, to cut the protein into peptides. So we know all these letters that appear there that I showed you before. We know this chain, and then we can track the changes. So what we usually do is this is a horse tooth. We have to be very careful to take out of the dentin because the dentin has other proteins like collagen, keratins, etc. And we don't want that in our sample because we are just focusing on the amelogenin. So this part, the outer part, is the enamel, so that's what we want to sample. And we need a very little amount. We cut a piece and we weight it. And then through all these different processes to cut the protein into peptides, then we have this little amount, and that's what goes into the nanoliquid chromatography and then mass spectrometry. And then after that, we will get some results. So the samples that I'm showing you here are coming from El Salt. This is a Middle Paleolithic site that is located in Spain, in the eastern part. It's about 700 meters of sea level, and it's an open-air site. And you can see very close to this site, there's another one that is also coming from the same period. And the site is located here, behind this very tall travertine deposit. And here it's called El Sal because El Sal means like the jump, because there's a waterfall there. When it rains a lot, this water still falls here down. And the site is located around here, and it's divided into 13 stratigraphic units. And all of them predominantly are dating from the Upper Pleistocene. Some Neanderthal remains have been found. They comprise 4 individuals, and there are 6 tooth that have been recovered. And the complete sequence is shown here. As I said, the dental remains are coming from this part, the upper part. And now we are working in this little layer here, that is the 10A. Because this layer is very rich in lithics and farmer remains, but also very interesting, also combustion structures. This is a very intense occupation of the site in this moment, so that's why we wanted to look at it in this time period. What were the activities of the humans in this site related to prey selection? So the faunal analysis of this concrete layer focus on medium-sized mammals, especially wild goats, deer, and horses. So this is the layer 10A. So one of the other taxa that is very abundant are the leporids, and carnivores are very minimal, very minimal presence. So what we are doing in Alacant in this layer, we are analyzing all these medium-sized mammals. We have samples from goats, deers, and horses, but today I only focusing on the horses to present here. And also, we know there is a very intense activity of prey hunting activities because there's a human substances marking, like breaking and cutting on the bones. So they want to go for the marrow. We have the breaking of large bones, but also we have cut marks. And this is happens in a lot of the samples, right? So they are actively pursuing this protein acquisition. So how this amelogenin works? So here what we have is the sequences that are in the databases, in the UniProt database. So what we usually do is we check for the differences in these changes in the amino acids. And this is the proteins that are sequenced. We have the males and the females, and then we check with our samples, right? The archaeological ones, and we have also those changes. Here we see in this middle region where these are changing. So we have the female, the chromosome X, but we also have the chromosome Y, and we have all this addition of different amino acids. So this is the result. I'm very preliminary, but from the 12 samples that we analyzed, 9 are males, 3 are female, and these 3 are males that we collected for modern reference because we wanted to have secure proteomes from the Y chromosome to compare it. So recovery of the protein is quite high and is well preserved, except for 2 samples that are a bit lower than 20% of the coverage of the protein. That would be also for maybe preservation issues. And that happens to maybe resolve some questions like about if they are mostly male that are going after the bigger prey, because the hunting groups are more specialized, are skilled, and maybe the human groups were bigger than we thought. And then we have to put this in their environmental context. And this technique, like a conclusion, is like really we can apply this technique to analyze dental remains or Pleistocene sites, push forward to our knowledge on the subsistence patterns of Neanderthal groups, also modern humans. Thank you.